Hi everyone, so second page of the mapping linked genes worksheet from last week's recitation. At the end of the first problem we were able to figure out the resulting chromosomes that would be produced if there was a crossing over event between the D and the R genes of the heterozygous parent. And what we're trying to figure out now is actually the relative distance between those D and R genes. So you take what you learned from the first problem and you apply it to this problem, but now we finally get some numbers to make a sense and an estimate of just how far apart these things might be. So to refresh your memory, what we had was two parents. We had one parent who had a heterozygous genotype, and we saw that their chromosomes looked like this. And then we had another parent whose genotype was homozygous recessive, so it looked like this. So we took that information, and you can imagine what we're doing is we're crossing these parents. So we have parent 1, we have parent 2, and they produce some offspring. One of the things we had been mentioning again and again and again was that you should find that you will always see certain things in the offspring that are indicative relative to the parental phenotypes and genotypes. So specifically what we mean by that is that there should be some numbers that jump out at you, and you can see that there are two numbers paired together that are relatively very large and two numbers paired together that are relatively very small. And the first thing that you should mention is, well, think about what the parents actually look like. And in this problem we've set up two physical characteristics, one for body color and one for nose color. In this case, the tan body is represented by the dominant D. So if you had a big D, you'd be tan. If you had a lowercase d, you would be dark. And likewise, if you had an uppercase R, you would have a black nose, and a lowercase r, a red nose. And I should say they're following a simple dominance model, so it's not that whether you have one or the other. So if you have simply one of the dominant uppercase letters, you will be that phenotype, and only in the instances in which, since these are diploid creatures, you have both recessive lowercase letters would you consider to have that phenotype. So applying that, think to what the parents had, and their parents are drawn below here. We see that in the heterozygous parent, obviously they have one of each of the dominant alleles, so we would expect parent 1 to look like tan and black phenotype. In parent 2, they'd be the recessive, so they would likely be dark and red. And if you think about what the products of their offspring are highlighted here, it doesn't exactly follow the saying that they should look more like their parents, because clearly what we see here is only one instance in which there's a dominant gene, and that would be the gene for body color. So it's tan and red, and I don't really have to rewrite this because we see that here. And nobody's tan and red. We have tan and black in the parents, and we have dark and red. So take caution when somebody's saying that there should be a large proportion of things that look like the parents. We're not talking about the phenotype necessarily, but we're talking about the specific combinations of D and R genes on a given chromosome. So a way of visualizing that is looking back to, when I've made a sort of a mess of it, but looking back to the parents. So what do the parents have? The parents had this combination of D and R on one chromosome, or this combination of D and R on another chromosome, or this combination of D and R on another chromosome, and obviously this one's identical. So do we see that in the resulting offspring? Well, uppercase D, lowercase r, look at this instance in the first place. Uppercase D, lowercase r, where else do we see it? That's it. And sure enough, there's a lot of that looks just like the parent. Go to the next one. Lowercase d, uppercase r. Where do we see that? It's not in the first one, not in the second one, not in the third one, but we do see it here in the fourth one. And sure enough, it's a big proportion of the overall offspring. And lastly, we could look at the lowercase d, lowercase r. And where do we see that? Well, we see it all over the place. But obviously in terms of the parental contribution from parent 2, we see it largely here and here. So why is this? Well, what we're trying to say here, and sort of address the question that I've now buried in green, but what we're really saying is, why are those types of combinations? Why is it that this and this combination seem to be so abundant, representing well over 90% of the overall offspring. Which, by the way, if we're thinking about crossing parents, this is kind of funny to me, because if we were thinking these as, like, I was thinking red nose, tan body, something like Rudolph, I don't think two deer are going to produce 7,000 offspring, but it just occurred to me that that might be kind of silly. But when we think about these classes, we see that those are representing pairs of genes, DNR genes, in combinations that look just like the parents. And that's really what we're trying to get at, is that the rate of recombination, no matter what it never produces more recombinant offspring than it does parental offspring. The upper limit to that is a 50-50 even distribution of recombinant and parental chromosomes. But in this case, we have far fewer recombinants. These are recombinants because they don't look like our parents. So we could skip. And same things again, same numbers, but we can address the second question. So why is it that there are lowercase dr and uppercase dr, lowercase dr? Why are those combinations of genotypes, those classes, more rare than the others? And basically saying, why are these numbers so small and why are these numbers so big? And sure enough, you just think back to what you had. The only way to get this kind of combination was if in the heterozygous parent we had initially this genotype. The only way to get something like this, and obviously in the other parent, the only thing there was the recessive. The only way you can get that combination, this parent can give only one of these chromosomes, and in either case they're going to give DR. But what can this parent give? The only way that they could ever give this lowercase d, lowercase r as well is what? If a crossing over event happened. So you'd need some sort of crossing over event to occur to produce this combination and provide that. But that's requiring recombination. It's not guaranteed the recombination is going to happen between those two genes specifically. So that makes it less likely to occur. So you're going to find that those genotypes are far less likely to occur than you would have otherwise. So the parental ones, much more likely to happen than the recombinant ones. The likelihood of that event happening would be, certainly the last question in calculating the frequency of these recombinant genes is very straightforward. All you do is you find what those two recombinant totals are, you add them together, and if we were to add that up, we'd find that 246 total over our 7,000 total progeny will equal a certain percent. And we're going to multiply this by 100 to express it as a percent, and it turns out that that is 3.5% total. And all we're saying there is that when we take the proportion of offspring who are recombinant, these ones that required recombination to occur to be produced, 3.5% of the time you're going to see recombination. And sort of think about what that might mean in terms of the relative distances. If you see a very low recombination event, it may be that those units, those genes, are physically located fairly close together. That is, if we see a lower abundance in the offspring that are the recombinant type, that means that crossing over isn't happening very often. And if crossing over isn't happening very often, that must mean that they're relatively close together. Hopefully that helps clear things up.